So let's start today's topic, or as you're gonna late, so those who will be joining late will be able to get their recording. So my name is Munaza Ada Khan, and I'm a PhD in analysis and engineering. And this workshop entitled uh, Electroanalytical Method for Biochemical Sensing mainly focuses on the electrochemical characterization technique that one can utilize for biomedical application or biochemical sensing stuff. So in days one decided range of topic, we couldn't finish it, so I'm going to go through it all. And there was a problem with the recording with the previous one, so I'm going to cover it up very quickly in today's. Presentation, and then I'll conclude the first part, which is actually which focuses on the fundamentals of the electrochemical method. So we start with electrochemical reaction, the electrolytic cell, electrokinetics, interfacial reaction classification, and we're not going to into the details of the potential sweep methods, but we'll just take a quick overview. Look at it. We'll discuss it in detail um, uh, last in tomorrow's topic. So electrochemical reactions, basically these are the chemical reactions which are potentially driven. So normally what happens, you apply specific potential to start a chemical reaction. And if you're characterizing a chemical reaction or a series of chemical reaction, so basically what you do is you sweep potential across a range to determine the nature of the chemical species which are present in your cell. So the main point to consider over here is you apply a potential, which is in the somewhere in the range where the formal potential of that chemical reaction or the chemical species it matches, and then it undergoes the reaction, and then this is how you start getting your IV response. So this is just a quick, recall of your high school chemistry. So basically, the electrochemical reaction it involves it can be reversible or irreversible redox process that involves a loss of electron in case of oxidation or gain of electron in case of reduction. So uh, this picture is this very visual picture is just to help you memorize what happens in the oxidation and what happens in the reduction process. So in electrochemical reaction, you have got this reactant in the solution, and then you have electrode which actually acts as your source or sink of electron, and then it provides electrons to your reacting species to undergo reduction reaction, and that's how you get your current response. So the electrochemical process, as expressed in this equation, it involves the transfer of charge across the interfacial region of metallic electrodes, which is basically a metallic electrode. And like I said earlier, it is basically either a source of electron or a sink for electron. So this reaction involves the transfer of charge species between the electrode surface and the solution phase. And this generates a potential difference between the solution and your electrode. So, an important parameter to consider over here is the area of your electrode, the geometry of your electrode you're using as a working electrode to apply potential and to probe the chemical species which are present in your um, cell. So these involve charged species whose energy depends on potential of the phase that such species are contained within. The phase is actually the aqueous phase, and the aqueous phase normally contains the probe species as well, which are meant to carry current. So this is a traditional um, configuration of the electrolytic cell, and normally you use a three electrode system. Um, you use a reference electrode, a working electrode, which is basically the electrode at which you scepter cross the potential, and you determine the interfacial reaction on your working electrode. And then you have a counter electrode, which is mainly platinum or a carbon-based electrode. So I'm trying to move this meeting control. Okay, yeah. So if you place it, basically, why do we need this three electrode configuration? Because if you're putting one electrode and working electrode, and then you're immersing another working electrode, and you're not maintaining the potential difference between them, what you're getting is a meaningless IV response. So basically, you use a reference electrode of saturated silver chloride. We also call it saturated calomel electrode, shorthand SCA, to keep the potential uh, difference, to keep the potential difference between them as low as possible or stable. So basically, see, there is a potential difference between the potential that's generated at the electrode and the potential that's which is due to the redox electroactive species you're using in your solution. And normally at equilibrium, no current is drawn from the cell, so the potential reaches a steady value, which is called equilibrium potential. And it depends on the relative concentration of the oxidized or the reduced species of your a reactant or the probe that you are using. So conventionally, the two different types of conventional reference electrode eh, are like a standard hydrogen electrode and this silver chloride saturated caramel electrode. So mainly people use SE based electrode, and this potential is due to the reference electrode, and it's normally kept constant and as minimum as possible to avoid interference into the electron response current response of your working electrode. So equilibrium chemistry helps you determine the thermodynamic parameters, for instance, entropy, free energy, equilibrium constant, etc. It's kind of a little dry and boring, but dynamic electrochemistry is our main interest, and it's normally employed in sensing, storage, and energy applications, energy generation applications, more specifically. So in this course, we'll be focusing more on dynamic electrochemistry. So in dynamic process, first a reactant diffuses towards the electrode interface, and then potential is applied, which is normally higher than the equilibrium potential to induce charge transfer process. And a current flows through this whole cell via electrolysis. And lastly, the current that is generated due to this electron transfer process is dependent on the flux of the electroactive species towards the electrode. And basically, electron transfer occurs through quantum tunneling effect, which normally occurs at a distance of 10 to 20 angstrom. So basically, this layer is 10 to 20 angstrom from the electrode. And some of the influencing factors are the type of the electrode material you're using, the geometry of your electrode, and the potential that you're using to probe the interfacial reaction, and the electron transfer kinetics, which is occurring at the interface. So if you consider it a dynamic process, it follows the Faraday's law, and the amount of the current generated is basically depends on the flux of the electroactive species at your interface. There's a legend of the equation. So A is your area, F is your Faraday constant, J is the flux, of the active species, which is mainly over here. If you're not using any other substrates or just the redox probes, then it's just the redox probe which is generating either due to one step or two-step process, depending on the choice of the probe you have decided to work with, and I is your current. So this is again the three electrode system electrolytic cell and its equivalent circuit. And like I said earlier, the potential across the reference electrode and working electrode is kept controlled to minimize the interference or to minimize the ohmic drop. And then your counter electrode is basically a non-reactive large surface area material, like I said earlier, normally platinum or carbon based. And the one more thing I would like to mention over here that the potential is capped minimum or close to zero at reference electrode because you don't want it to get polarized. So we want to polarize only our working electrode, to beyond its polarization potential, so that the current flows across the circuit. So electric double layer is formed at the interface, and it's classified into four main types: the compact layer, the diffuse layer, the diffusion layer, and the bulk solution. So the closest one is the compact layer, and it's actually the layer which is less than 1 angstrom, and it's here the electron transfer process takes place. So this is a close-up or zoomed perspective of the compact layer, and you can see over here an inner Helmholtz plane and an outer Helmholtz plane, and cationic, specific cationic and anionic species are absorbed absorbed inside this layer. So basically, the response that you're recording is due to the diffusion of these electroactive species, be it cation or anion, from your, chemical reaction. So you're measuring this one. And then you have this diffuse layer where diffusion is occurring, and Josh transfer process is very active in your compact layer, and the process of diffusion is taking place. It's around 1 nanometer. This is called a diffuse layer. And then next to diffuse layer is actually a diffusion layer where your mass transfer process is taking place. It normally ranges in microns. So the mass transport process basically occurs through three different ways. The first one is diffusion, which occurs, normally at your compact layer, and then there is convection and migration, which normally happens, and the bulk solution. So in the stagnant solution, where your solution is stagnant, normally diffusion occurs at the interface, and it has a specific range, like this is the delta across which below which it's going to generate the response of your electrolytic cell. And if the distance of the species extends beyond the delta or the critical distance, so it won't generate the whole process is the diffusion control, and it won't contribute in the generation of current generation process. So this is NERNST equation that defines the process of diffusion, at the interface. And this is your concentration gradient. This is your diffusion coefficient. And this is your potential gradient. So it takes into account the distance from the electrode, and then it on the basis of it, concentration gradient and potential gradient, the both are taken into consideration for a general bulk solution mass transport. And In stagnant layer, only fixed layer is applicable because in this layer, only the concentration gradient is basically defining your flux, which is basically the concentration of your electroactive species at the interface multiplied by its diffusion coefficient. So as said earlier, electrochemical reactions are potentially driven, and you apply some sort of electrochemical pressure to start a reaction. So recalling potential is a energy required to move charged particles. It's normally measured in joules per coulomb. So when the electrode potential is above the Fermi level, and that is higher than the LOMO. So, you have got two different bands. Highly occupied molecular orbital and lowest unoccupied molecular orbital. So when your Fermi level is below, LOMO, there's no charge transfer process. But when your Fermi level is higher than your LOMO, so in this case particularly, so the Fermi energy level is higher than LOMO, then there will be a transfer of electronic species, and it will uh, the current will flow through your circuit. So basically, this is how, by varying potential, you generate the response through a chemical reaction. So normally, so this is basically a plot of free energy, Gibbs free energy against the reaction coordinate. And recalling that cathode is the electrode that supply electrons for a reduction process, and anode does the opposite. So the rate law for net process is this one. This defines the rate constant of your electrochemical process that's taking place inside the cell, and they are dependent. It's dependent on the potential, and when you apply the voltage, your free energy, what actually when you apply the potential, what actually it does is it increases the free energy. So the solid line over here, and as free energy is actually a function of the applied potential. So the solid line, the dashed line over here, shows the energy of the reactant where the potential is zero. But as you increase as you apply the potential, the energy of the system increases, and it's dependent on the transfer coefficient as well as the number of the electron that are being transferred, and the energy barrier you see over here decreases as you apply the potential. So your reactant easily undergoes into transition state, and then your product is formed. So it sort of decreases the amount of the activation energy that is required to transform your reactant into, into product. So the geometry of the electrode affects the type of, the IV response you generate. So this is a cyclic voltammogram where you apply a forward potential and a reverse potential to prove the electrochemical reaction. So in case if you're using electrode, which is a macro in size, so what happens, the electrolysis across the entire electrode surface it occurs through planar diffusion process. So normally at edges, you have this insulating material, and you get a convergent response, convergent diffusion response around the edges. But in case of macro electrode, this edge effect is normally, inundated by the planar diffusion response, which is taking place across the whole, entire surface of the electrode. And you get this nice reversible voltammogram with symmetry. So it's very nicely shaped, and you can easily differentiate the reversibility or irreversibility of the chemical reaction that's taking place. I'm going to discuss it in detail in potential sweep methods. So, so basically, planar diffusion is predominantly observed when you use macro electrode. But when the size of the electrode is reduced to microns, so the edge effect becomes more prominent, and you get a sigmoidal peak uh, of cyclic voltammetry in voltammogram, and convergent diffusion over here becomes more prominent, and it has benefits in terms of when you want to improve the mass transfer process. So normally when you're using microelectrode, uh, what you're doing is you're increasing the surface area. So when you're increasing the surface area, now you have to consider something over here. The surface area of the microelectrode is high compared to the macroelectrode, right? So the edge effect is more pronounced, which resulted in the loss of the shape of the cyclic voltammogram. So and it actually this approach, the microelectrode approach, is normally it's been used in nanoscience engineering research studies that involve biosensing or design of the biosensors, because it improves the IV response, and since the edges have this convergent diffusion process, so in case of sensor, you are actually attaching, you are coating your electrode. In a simple word, I should say that you're coating your electrode with the whole complex that helps you detect the analyte, or that makes it a sensor. So basically, it actually further enhances that convergent diffusion response, which results in a loss of the strip. So the size effect is supposed to be taken into consideration when you're reducing. Size effect for IV response should be considered when you are reducing or changing the shape of your electrode. And now coming to the part where we're going to discuss interfacial reaction kinetics. So when you are probing some chemical reaction, or let's say you have modified your electrode for XYZ application. Over here, I'm going to code like for sensing application. And let's say this is your electrode, and this is the detecting species that binds to your electrode. Once you swept across the potential according to your desired range, whatever it is, and this is specific detecting species will generate current response at the potential where it's undergoing the redox reaction. And and this you're going to record in the form of the cyclic voltammogram, right? So if let's say if it's a one step reversible electron transfer process, so you're going to get a reduction peak and 1K30 peak, considering it an IOPEC representation. And this confirms the presence or at that specific potential. The peak potential will help you determine the presence or the binding of this specific detecting species on your electrode. So, so there are different types of interfacial reactions. It can be heterogeneous, homogeneous, one step, two step, or multi step electron transfer process. So this you can easily probe through a variety of technique and potential sweep technique. But to drive the current throughout your solution, you actually have to use a redox species or a redox probe. So normally, it can be a positive, it can be a negative, depending on the type of the reaction or the detecting species that you're using. So I quote here example of my work. So my the double layer at the electrode that was formed in my case, it was actually positively charged. So I had to use a negative redox probe inside, which is actually dissolved in the solution to drive the current, um, so that it maintains the potential across the circuit. So So in case if you have this whole, the net charge of this whole detecting species is negative, you're going to use a rubidium based, or some kind of positive redox probe. So depending on the probe that you are using and depending on the type of the interfacial reaction which is occurring at the interface of your electrode, there are different types of interfacial reaction. So this is from the book of the Alan Jabard. Yeah. So for further details, you can actually find the book. So what happens if you take a look at this one, case one, so we call the reaction of the redox probe that you're using. It's normally called coupled reaction, and the reaction that is taking place at your electrode interface, it's called as electrode reaction. So we represent a couple electrode with C and the electrode that is taking a chemical reaction that's taking place at electrode by E. So there are different types of it. So C and E, and it's really important which comes first. So if C comes first, it means the first your redox probe undergoes the reduction or redox reaction, whether it's the reduction or oxidation, whatever it is, whatever you choose to work with. So if it precedes the electrode reaction, this is how you're going to write it, and R stands for reversible or irreversible. So so this is a coupled reaction, and this is your electrode reaction, and in this case, the both are reversible. So this is how you're going to define it. Your coupled reaction occurs first inside the solution, and then the reaction takes place at the electrode interface. And this also defines the shape of the IV response that you get. So IV peaks will vary according to the type of the probe that you are working or the electrode modification you have introduced, which has resulted in a range of different types of reactions. So it can be one step electron transfer. So in case if it's one step electron transfer which is reversible, you are going to represent it by ER. So in first case it's both reversible, and in second case your coupled reaction is occurs first, and then your electrode reaction occurs, but it's irreversible, right? So you're going to get one peak if you're using forward or reverse potential, so depending on the technique that you're working with. And the third case is in third case, I think it's the font is very small, so I think the electrode reaction occurs first, right? And it's reversible. And then your coupled reaction occurs and it's also reversible. The both are reversible. It's very similar to this one, but this time, basically, the reaction at your electrode occurs first, and then your coupled reaction occurs. It means the redox species undergoes a redox reaction. Your probe undergoes redox reaction after the electrode after the electrode interface reaction. And in the fourth case, the coupled reaction is actually irreversible. So after this reaction takes place at the electrode interface, this reaction occurs in your solution, and this is irreversible. So the list, the combination goes on, and depending on the type of the reaction you're working with, their diffusion equation varies, and their current IV response varies. So this is a few more examples. And sometimes the electron transfer process is actually multi-step. So in this case, in this particular case, uh, two reversible electron transfer takes place, and the both are reversible. You see, so it means you're expecting a double peak in case if you're working with a cyclic voltammogram. It means that you're expecting double peak in your cyclic voltammogram. So I'll be covering this in potential sweep methods. This is all for today. And let me see if participants have got questions related to the fundamental concepts of electrochemistry. Okay, so we don't have questions so far. So we wrap up this section, and